Welcome to the BioTech Whisperer channel. Our topic today is on the perspectives surrounding first lab-grown meat burger. If you are new here, we are a group of retired professors sharing bite-sized videos. Let's continue with our topic. Building a $325,000 burger. In 2013, the world's first burger was made from cultured meat produced from bovine stem cells, but this cost many thousands of dollars and involved the combining of at least 10,000 individual muscle strips. As a gastronomic delicacy, the 5-ounce hamburger, assembled from tiny bits of beef muscle tissue grown in a laboratory and to be cooked and eaten at an event in London, perhaps in a few weeks, is meant to show the world, including potential sources of research funds, that so-called in-vitro meat, or cultured meat, is a reality. Although muscle strips and cultured meat products have been produced, there are still many technical difficulties that will need to be resolved for the large-scale production of products acceptable as alternatives to conventional meat for discerning consumers. Cultured muscle has been produced by taking skeletal muscle stem cells, myosatellite cells, from live animals and inducing them to grow and differentiate to form muscle fibers in vitro, but the development of methods to do this in a commercially viable way has yet to be achieved. Cultured meat, which is also known as synthetic or in vitro meat, is seen as having a number of advantages relative to conventional meat in terms of efficiency of resource use, land, energy, and water, lower greenhouse gas production, better animal welfare, and in the ability to manipulate the nutrient composition of the product. Terminologies. The term cultured meat will be used here as it seems to be the most widely used and accepted term, but alternative terms used for the same product include synthetic meat, in vitro meat, and sometimes artificial meat. These are generally interchangeable, but are distinct from the term simulated meat, which encompasses products that are similar in some respects to meat, but are made from non-animal proteins, such as those from plants, especially soybean, and fungi. Growing muscle tissue in culture from animal stem cells to produce meat theoretically eliminates the need to sacrifice animals. So-called cultured or synthetic or in vitro meat could in theory be constructed with different characteristics and be produced faster and more efficiently than traditional meat. The technique to generate cultured muscle tissues from stem cells was described long ago, but has not yet been developed for the commercial production of cultured meat products. The technology is at an early stage and prerequisites of implementation include a reasonably high level of consumer acceptance and the development of commercially viable means of large-scale production. Discussion on advances. Recent advancements in tissue culture techniques suggest that production may be economically feasible, provided it has physical properties in terms of color, flavor, aroma, texture and palatability that are comparable to conventional meat. Although considerable progress has been made during recent years, important issues remain to be resolved, including the characterization of social and ethical constraints, the fine-tuning of culture conditions, and the development of culture media that are cost-effective and free of animal products. Consumer acceptance and confidence in in-vitro produced cultured meat might be a significant impediment that hinders the marketing process. For instance, consumers, however, may be cautious about accepting such products due to perceptions of unnaturalness and artificialness. Although benchtop bioreactors have so far been quite successful in meeting current demand for cultured meat, as the demand increases, greater innovation is warranted in bioreactor design. In addition, most commercial bioreactors are designed for growing suspension cells or cells anchored onto microcarriers, and not scaffold materials with thick tissue layers. This makes it challenging to directly adapt available designs for cultured meat production due to system incompatibility like diffusion limits. The culture media surrounding the growing cells and fibers in culture must contain not only adequate levels of all necessary nutrients and oxygen, but also growth factors and bioactive compounds required for normal muscle development. Currently some of these are obtained from animal tissues, but eventually it would be desirable for the media to be free of all animal products. Growth in vitro of a single layer of myocytes and muscle fibers on a base of collagen fibers has been achieved, but the formation of stick-like three-dimensional structures will require a 3D framework or scaffold and a means of ensuring that every cell/fiber has a continuous and adequate supply of nutrients and oxygen, as well as a means of removing waste products such as carbon dioxide. It is likely that the initial cultured meat products will simulate processed meat items, such as meats with limited structural requirements in terms of a meat-like texture. The production of steak or roast-like products, however, likely requires significant further developmental research concerning scaffolds, circulatory systems, and the creation of key quality attributes, such as flavor and tenderness. If successfully produced, it is possible that cultured meat products could play a useful complementary role alongside conventional meat products in meeting predicted increases in the global demand for meat. Visions to be met. Whilst it is envisioned that cultured meat has the potential to address all of the externalities associated with conventional industrial meat production, from environmental impacts and animal welfare considerations to public health risks associated with zoonotic disease and antibiotic resistance, while ensuring a scalable and secure production system to meet the anticipated growth in demand for animal protein. The extent to which they constitute competitors of conventional meat remains to be seen. Forward perspectives. Several key challenges remain in producing cultured meat, including access to proprietary cell lines, high raw material cost, animal source nutrients, and limited manufacturing scale. Despite this, immense progress has been made over the last decade. It is too soon to accurately assess the readiness with which consumers will accept cultured meat products. The real test will be when such products are on the market. Cultured meat provides a hope that our society can become less reliant on animals for meat, thus reducing the environmental and health impact of animal farming. There are still major scientific challenges, including developing quality cell lines, lowering the cost of growth media components, and designing bioreactors for growing thick tissue layers before cultured meat can become a common food product. Nevertheless, there is room for optimism.